We're presenting at the ASH meeting this year our findings on immunoglobulin lambda translocations portending to poor outcomes in multiple myeloma. For this study, we took advantage of the MMRF COMPASS study, particularly the large set of whole genome sequencing to study the structural variations in myeloma and which ones may result or associated with a poor outcome. And in taking on this large project, we found that translocations to the immunoglobulin lambda locus on chromosome 22, the patients who had that did poorly compared to other patients in the study. This is over 900 patient samples that we analyzed, and there were about 10% of them had immunoglobulin or IgL translocations. Of those, what was really interesting was 70% of them were patients who have hyperdiploid disease. And this is something that we typically think of as a good prognostic marker or standard risk disease. So these patients are being underdiagnosed. They have actually a high-risk disease and they're being told that they have a standard risk disease. So it's a new marker that we have not used before. And so we then went on to determine why these patients were doing poorly. And it was a little bit surprising that we couldn't find a lot of other genetic alterations that were different. But one thing we noted was that the translocation, when we did some chromatin work with our colleagues at the University of Calgary, that Icaros was bound very highly to the IgL enhancer. And this is one of the targets of the IMiD drugs like lenalidomide. And so we find that the patients who have this translocation do not respond well to these IMiDs, and that's why they're doing poorly.